So during the coffee break, the GASBOR modeling process has finished. So here you see on the left the original high-resolution structure. This one is a high-resolution structure solved by X-ray crystallography, and this is a higher resolution ab initio model that came from the program GASBOR. As you can see, the overall fit is very well. However, if you have a good look at the models, you can see that the GASBOR model has the wrong handness, so it's rotated the wrong way compared to the real structure. So it's like the mirror image of the structure that was solved by the X-ray crystallography. So this is because we do not see handness in SAXS, and if you would repeat the process of the GASBOR modeling, you will get very similar models, but however they could be mirror images of each other. And in SAXS there is no way to see which one is correct. So, however, usually if you do not have a way of resolving the handness, you just ignore that, and any journal would accept the SAXS model with the note that the handness is not known. So let's continue with advanced modeling. So I will tell you in this talk how to validate high-resolution structures against solution scattering data. And we will have a good look at a process called rigid body modeling. And I will briefly tell you about how we can deal with oligomeric mixtures and with flexible proteins. So we estimated the overall parameters of our particle of interest and maybe even got an ab initio shape. So these two steps are essential and they do not require any additional information. However, SAXS becomes way more powerful when it is combined with complementary methods. For example, if we have a high-resolution structure from NMR or from X-ray crystallography, it is possible to validate the structure in solution. So basically what we can do is we can compute small angle scattering from this high-resolution model and to see how good or how bad it fits the experimental data. So computing the scattering directly from the atomic model is a fairly straightforward procedure. I will not go into mathematical details, but I must mention that computing the model in solution, the scattering from the model in solution, is different from just computing the theoretical scattering from an atomic model. So this is because in solution, the protein or whatever other particle will be surrounded by the hydration shell. Hydration shell means that the orientation of the solvent molecules close to the surface of the protein will be different. So basically water will have a slightly different electron density. And besides, there is excluded volume, so the volume inside the protein where the solvent cannot get in. So this volume will also scatter differently from volume filled with the solvent. So basically you have to, from the atomic scattering, you have to subtract the scattering from the excluded volume and add the scattering from the hydration layer. So besides using the spherical harmonics approach, it is possible to perform all this analytically. So, and this is a key to all further modeling where high-resolution models are involved. So this permits to make programs that will give you an answer in a reasonable time. And the most popular approach to modeling is called rigid body modeling. So rigid body means that we see our model as a number of rigid objects that are not flexible, but they're rigid, and we can combine them together to see if this model composed from rigid submodels produces scattering that could fit the experimental data. So typically, if you ended up with SAXS while studying your protein, you want to have information about either a complex or a multi-domain proteins, and there is information from the individual macromolecules already available. So either the like single domains of the protein were solved by other methods, or you can find a structure of a domain which has a similar amino acid sequence. So, and large constructs, they are very difficult to study using, so they are too big for NMR and difficult to crystallize. So this way you can try different superimpositions of the individual subunits to see if the complex would produce scattering that would fit the experimental data. So in this example, the program SAXS is trying to superimpose 5 domains of the protein in a way that the overall structure would fit the experimental data. So here we basically took the protein, cut it into 5 pieces, crystallized or whatever, solved the structures of individual components, and then these individual components, subdomains, are connected to each other in a chain, and then the program finds such a position of this chain that the scattering fits the experimental data. So obviously this approach implemented in the program called SAXS can take these factors into account, that the model must be interconnected and there should not be any clashes. So the individual subunits should not touch each other too much. Besides, it is possible to apply symmetry if we know the symmetry in advance. And there is also, it is possible to use information not just from one measurement of a complex, but here in this case we have a complex with symmetry P6, but under certain conditions we can make sure that this complex dissociates, and we measure just the subcomplexes with symmetries P1. So we can plan our experiment in a way that we measure both this complex as a monomer and as a hexamer, and use both data to build a model that where the overall model is a hexamer, but the individual monomer would fit the data from the monomer. Besides, sometimes the symmetry is slightly more complicated, like here we have the one subunit of the complex has a symmetry P6, the red one, and the other one has a symmetry P3. So it is possible to add such restrictions to the program. Besides, it is possible in rigid body modeling to use various information from other methods, such as if we know where exactly do our subunits contact, we can, for example, it's possible to obtain such information from NMR or from other methods, then we can say that these two subunits have to have a contact here. It is possible to measure distances between certain amino acid of protein 1 and certain amino acid of protein 2. And this information can be taken into account. And it is possible to measure the relative orientation of the subunits. And SAXS is typically not very sensitive in this account, but you can get this information from NMR. Besides, you can, if you have a large complex, you can measure subcomplexes and use this data together with the process of resolving the larger complex. All this helps to reduce ambiguity, and all this helps to stabilize your solution. If you have a lot of additional information, you will get very stable solutions from multiple runs. Besides, you can exclude part of the information or use a part of the information to validate your models. So I mentioned subcomplexes. This is assuming that the individual components of the complex do not change the conformation when they are alone, not in the complex. If we make this assumption, which is sometimes the case, we can construct various subunits, subcomplexes, and measure them. So like in this case, we have a complex of 4 subunits, and we could remove one subunit or another to measure a subcomplex of 3 subunits or smaller constructs of 2 subunits. If the individual components change the conformation when they are in a complex, then small angle neutron scattering could help you to resolve that problem. However, it will be the same program called SASREF that could help you to construct such a complex of 4 subunits that the subcomplexes would fit the additional data. So this program has an online interface. Basically what you need is the data. You need to say how many curves you measured from different constructs and how many subunits you have and what the overall symmetry is. I will demonstrate you the interface later. So rigid body modeling is good when we have information about all the components of our complex of interest. However, very often there is only partial information available, and it is possible to add this partial information to the big picture. So sometimes flexible or semi-flexible loops or whole domains are not resolved in high-resolution models, or they are removed in order to make the protein crystallize. So, however, we can model these removed parts or these missing parts using the program called Bunch. So again, it will use the available high-resolution information and will reconstruct sort of a chain, similar like GASBOR does it, using beads instead of residues. So In this program, you can also restrict the positions or just the orientations of the domains from which structure is available. And it will reconstruct the possible conformations of the missing linkers. Here you can also do quite many things. For example, you can fit multiple scattering curves from partial constructs. For example, if you have a 3-domain protein, you can also make shorter constructs like just domain 1 and domain 2, or like domain 2 and domain 3, and measure that as well. You can introduce symmetry if you know the symmetry of the overall construct. And you can fix some of the domains in order if their relative orientation is known. So contacts are possible. The program will also check that there are no steric clashes. However, this approach is applicable only to single chain proteins. So you cannot do complexes with it. Luckily enough, we created another program called Coral that combines the approaches of SASREF and Bunch. And it permits to model multi-domain protein complexes against multiple data sets if the multiple data sets are applicable. Instead of the dummy residues, it uses a library of short loops. It's a fairly big library, and it will add these loops that are generated from using the statistics data from the proteins, using these loops instead of these dummy residue chains. So, however, when you do modeling against SAXS data, you need to keep several things in mind. So first of all, SAXS is a low-resolution method. Even when you use high-resolution models, when you do SAXS modeling, the resulting model is still low-resolution, even if it looks different. Besides, I already mentioned it a couple of times, there might be more than one shape or more than one way to superimpose the available structures that would produce an identical scattering pattern. And you need additional information to resolve this ambiguity. So even when you think you have a lot of additional information that reduces the ambiguity, with all these restrictions, one must cross-validate SAXS models against all available biochemical, biophysical information. If there is something that you can do, you can measure with your protein that would help you to validate your SAXS model, please measure it. And last but not least, the sample is never perfect. There is no such thing as an ideal solution of the sample. There is always something else in the sample. The sample is never perfectly monodisperse, and the sample is never perfectly homogeneous. So you have to keep that in mind. I explained you how to minimize these effects. However, if you think that the final models are wrong, the first thing to do is to blame the sample, not the programs. So all these approaches are applicable to relatively rigid proteins or other macromolecules. And we assumed that the solution was perfect, that is monodisperse and homogeneous. However, it is also possible to work with non-monodisperse solutions, namely with oligomeric mixtures. So if you have a mixture of more than one component, then it is, and you know what components you have, it is possible to estimate how much component 1 and how much component 2 you have in your mixture. For example, let's assume we have a monomer-dimer mixture. If we have a structure of the dimer, we can compute the theoretical scattering from it, and we can compute the theoretical scattering of the monomer. So then you can feed this to theoretical curves and your experimental data to the program called Oligomeric, and this program will tell you, so first of all it will try to fit the experimental data with a mixture of the monomers and dimers in this case. And it will tell you if it is possible at all to fit your data with these monomers and dimers. And if it is possible, then you will also get the volume fractions of monomers and volume fractions of dimers. This comes in very handy because you can also, like if you have the models of trimers and tetramers, you can add them as well. And the program will tell you that you do not have any trimers there, just monomers, dimers, and tetramers. And in which quantities. Besides, you can vary the parameters, for example, the buffer conditions, to see if the amount of the oligomers changes or not. So here we have an example of a dimer that under various conditions exists as either a monomer or a monomer-dimer mixture. So, and the structure of a dimer here was reconstructed using the monomer and rigid body modeling with the monomer. So you can do also more sophisticated things. So as I mentioned, it can be more than two oligomers and quite a few conditions. Another case of non-monodisperse solution would be case of a flexible system. So if you have a flexible protein, it's not really monodisperse in solution. So let's assume we have this two-domain protein connected by a flexible linker, and in solution it actually exists as quite a few different conformations. So basically we cannot solve the structure of this protein. So this protein exists in various conformations, so there is no one single model that could represent this protein in solution. However, we can analyze what kind of flexibility we have there. So we can analyze it quantitatively. And the way of doing that is called the ensemble optimization method. The idea of the method is that we generate multiple conformations. Typically, it's something like 10,000 conformations. So there is a program where you can feed the input data, like in this case, domain 1, domain 2, the overall sequence, and the program will generate 10, 20,000 of random models that would still make sense, but they would be random. So the distances between the domains will be different, and the conformation of the chain in between will be different. From each such model, we can calculate the theoretical scattering. And then what we can do is we can start actually a genetic algorithm, an algorithm that would pick various groups of structures and try to fit the experimental data using a mixture of the structures. So from this pool of thousands of models, it would choose a small ensemble, like 10, 20 models, that would describe that would fit the experimental data. Again, This way, we do not get a model or even a number of models. What we do get is from this solution. What we can do there is we can calculate the theoretical radius of gyration from each of these curves, and then we can plot the distribution of how the radius of gyration changes and compare it to the distribution of the radius of gyration of the overall pool of 10,000 models. So here in gray I have the RG distribution of the overall pool that shows how different the models can be theoretically, and in red I have the distribution for the selected models, and from the how broad or how narrow this peak is, it is a clear description how flexible our protein of interest is. So this approach also has a web interface. As you can see here, you have to provide the experimental data, the units of the experimental data. You have to upload the sequence of the full protein. You can choose between two different ways of generating the flexible parts of the proteins. They can be either random coil, like in intrinsically unfolded proteins, or they could more follow the native-like ways how it behaves inside the protein, a folded one. You can provide the number of domains that you use in that you have available from your model, and the program will do the rest for you. So all this is available from the SAXS package. That's a package that the group where I work in develops and maintains. There are also some other programs that you can use. Feel free to experiment with them. However, for solution scattering for biological macromolecules, I guess SAXS is a very good point to start with. So, to summarize the modeling part, we have various options depending on what kind of information we have. So if there is nothing known about the protein of interest or the particle of interest, we can always do the ab initio reconstruction. If everything is known, like you have a high resolution structure from whatever method, you can still validate this structure to see if in solution it looks the same as in the crystal. Besides, you may also check if in solution it is a mixture of different oligomers. If you have partial high-resolution structure, for example, high-resolution structure of individual subunits, then you can do the rigid body modeling. And if you have an incomplete structure of your protein, you can add the missing linkers using the program Bunch or Coral. And besides, SAXS is probably the only method where you can really characterize unfolded proteins. And unfolded proteins are about 30% of all proteins. So if you have questions and I'm already not around, please feel free to go to the SAXS forum. So if you just Google SAXS forum, you probably will find it. Just register there, it's very simple, and you can ask questions, or if you don't want to register, you can just browse through it. Most probably your question was already asked, and either members of our group or other users will try to help you. So, and I think I will finish the theoretical part for today. Thank you very much for your attention. Questions? the most interesting modeling part, and you don't have any questions. I guess that was a little bit too much information for one day. However, let's proceed with, with our practical demonstration. So, remember, we also measured the complex, and we even decided that this is actually a complex because the molecular weight would fit the molecular weight from the complex. So we already have the ab initio parts of it. So for example, if we open this model and point B, so we could already speculate how do they form a complex. So knowing that the radius of gyration didn't change, would you assume that they form a complex like this, or rather like this? So who is for this type of complex? Okay, either you are asleep or you learned everything very well. So who would prefer this type of complex? Okay, everybody is still awake. Good. So this is already, we do not have to do any modeling. So this is common sense that you always need to remember to apply to the questions that you would like to get answered using small angle scattering. And I must say this is very important. Before you do any modeling, I think in any structural biology, you need to ask yourself, what is the question I would like to answer? Because typically people come to us and say like, I want to know the structure of this protein. And it's like, yeah, go crystallize it. I cannot crystallize it. Give me the structure. Yeah, so I think I could demonstrate to what extent we could give a structure. However, typically the question is more sophisticated. For example, how exactly is the complex formed? Does the complex hold under these conditions? Is my protein flexible? Does my protein change conformation upon complex binding? These are valid questions that you can ask. So, however, here we can use a modeling program called called SAXS. So let us go through the steps. So the first step, we need to describe our problem. So we have one curve, that's the curve from the complex that we're going to use. And we have two subunits. Okay, I forgot to tell you that during our SAXS experiment and data processing, somebody managed to crystallize the protein A and protein B, and these are their high resolution structures. So we do have them available. So, and we have two subunits, one data curve, so we're going to use only the curve from the overall complex, and we do not have any symmetry here. So at the next step, let's give some ID. Yeah. Oh, damn it. So, and we have to choose our experimental file, which is the data from the complex. So the D2O fraction question is for the case of the neutron scattering, so we just skip it. The symmetry for this curve is still P1, and the angular units are reverse nanometers. So the fraction to fit is how much of the curve to fit. So 1 means fit the whole curve, 0.5 would mean fit only the low angle half of the curve. So let's fit the whole curve. So the weight makes sense if you have multiple data sets, and you can say pay more attention to the data set from the overall complex rather than to the data set from the subcomplex, for example. Here we have only one data set, so we just leave it as 1. And the contact question, the question is the constant question. Means that if you want to use a constant, the program may subtract the constant from the data in order to make the fit better. So usually you say no, you don't want to use a constant. However, with a constant, the fit will look slightly better. So the reason for a constant I already mentioned. It is because when you do buffer subtraction from, especially from large anisometric particles, the signal from the buffer, it's not exactly what we want to subtract. So, and you can compensate against it using a constant. However, it will only subtract the constant. It will not attempt to add a constant, for example. And there are certain limits which would make sense to compensate against this buffer subtraction. Usually, just don't use a constant unless you're really desperate. So, and then we choose the protein A, high resolution structure, protein B. So, and it asks if it should shift the structure. So shift means that before starting, the program will put the structure into the center of coordinates, so that the center of masses of each subunit will be at 0, 0, 0. This is done because if you shift the structure from the center of coordinates, the calculation of the scattering might become not that correct. So usually you say yes, and the program then will put your structures apart in order that they do not have steric clashes. So besides, we have the option to constrain the movements. So for example, we can fix one of the subunits, then this subunit will be, its position into space will not be changed. Or we can restrict the movements of the subunits. For example, if you have relative orientation of the subunits from NMR RDCs, then you can prearrange your subunits and then tell the program that they shouldn't be rotated against each other. So let's fix the first subunit and do not touch the second one. Actually, we could say that both should not be fixed. And yeah, in this case, one subunit will be moved to the center of coordinates and the other one will be moved around it. Probably it's better to say that they both should not be fixed. If you fixed one of them, it's slightly faster. So, and we could provide some contact conditions. So let's do that. However, in our case, we pretend that we do not know any contact conditions, so we do not know how the files are interconnected. So here's a curve subunit correspondence. That's again for cases where you have many subunits and many curves from the subcomplexes. Here we just leave it 0. And let's look what the contact conditions are. So here we can select first subunit and the second subunit, so we have only 2. And we can say here that subunit 1 from residue whatever till residue whatever should contact with the first subunit, for example, of first residue of the next subunit. So that means that something between residues 144 and 194 should contact with the residue B minus 3 in this subunit. So sometimes you just have this information and you can say that you should have a contact here. So, however, we do not have this information, we can restrict it saying that these two subunits should contact. So if we do not put this restriction, most probably it will work. But here I just select first to last residues, saying that they should contact somewhere. So typically, if you do not put this restriction, the program will still come up with a with a model, but the model might be disconnected if that would yield a better fit. So, and you say that the distance should be, I don't know, 5 angstrom, 10 angstrom, and you click submit. And after a few hours, you will get a model of the complex. Luckily enough, I have done this before. So, and this is how the result will look like. So here in green we have our experimental data, and in red we have the theoretical scattering from the complex. And here we have the PDB file with the complex. Save it. And this is the complex. So this complex was, I think, actually crystallized as a complex. So I have the real complex here, and it is PDB ID 106S. Okay, let's display it on the backbone. So this is the complex how it was crystallized, and this is the complex that was constructed using SAXS data from the complex and the two subunits. So as you can see, the overall result is not entirely incorrect, and this the complex constructed by SAXS was constructed without any prior knowledge how the subunits should be oriented. So this superimposition was found by like trial and error in a way that the scattering of the overall complex is fitting the experimental data. So the position of the subunit is actually correct. However, the orientation is not. So one way of dealing with that problem would be to repeat this modeling process multiple times and to see if there is some preferable orientation of the smaller protein. However, it's relatively slow and small and globular, and it's probably there will be no preferable orientation. So the proper way of dealing with this problem is to find the relative orientation of the two proteins using some other methods. So I hope I gave you enough practical information of how to use small angle X-ray scattering data if you want to model complexes. And I am ready to answer your questions. Yes, please?
For the contact information, you showed that you can define residue pairs.
Mm-hmm.
But that's just one entry, right? Say, for example, if I have biological data, so I know one spot is interacting with the other side, but I would like to have, say, a few more kind of restraints. Is that possible?
So yeah, basically, so let me see if this works here. Yeah, let's just redo it. What's wrong? So here we have two subunits, and we can select not, so if we select just here that the first subunit, first residue should contact with the first residue and give some distance in angstroms, that would mean that really the program will measure the distance from residue to residue. But we can say that from the first residue, so till this residue, and the program will take any residue in between, so whichever is close, like at the correct distance to the residue mentioned here. So you can add ranges, and if one of the residues in this range would be at this distance to another residue in the other range, then the penalty would be 0. And like this will be considered done. Besides, you can add an alternative. So, for example, you can add like either this or the last residue should be at this distance to whatever another residue. So, and then the program will use either one condition or another condition, because sometimes there is a certain ambiguity in this information and you need to add alternatives. Yeah. Does that answer your question?
Yeah, so what's the difference between add condition and add alternative?
So add condition, we can say that if if we had more than two subunits, then that would describe distances under another condition, other subunits. Yes, please.
You've talked about predicting linker sequence, the shape of the linker domain, right? If two domains, if we do not know the shape of the linker, what if I submit a false case? Will the server still predict a linker sequence between them?
So if you want to add a missing linker, it will come up with something. So if you use the program bunch, it works well only for linkers of certain lengths. So if you have a 100 kilodalton protein and you know only the structure for half of it, it will not reconstruct you the other half by folding this linker. It will give you something that will obviously look like nonsense, and maybe even the fit will be bad. So it is important always to check before checking the model, to check if this model fits the data at all. So this program called Coral, the result might be slightly better, but it has inside the program a certain limit for the linker that it can reconstruct. So it will not allow you to reconstruct a longer one. So, good. Are you guys still alive? So I can give you a very small presentation about automation of data collection. So this is more like data collection and analysis. This is more what I do. So beamlines, where you can collect SAXS data, can be very different. So when you are at the beamline, so I started doing SAXS like in 2006, and everything everywhere in the world looked like this. So this is actually the beamline at the old SAXS beamline in Lund in Sweden. I'm not sure if this beamline still exists. It was used for solution scattering and for powder diffraction. And back then it was a second generation synchrotron with quite a weak beam. But most importantly, the process of loading the sample would not be automatic. So you actually had to come inside the hutch every time, and using a syringe, you would first wash the sample cell. Then you would dry it with compressed air. And then you very carefully load the sample, observing the cell from the top. So after that, you leave the hutch, do the interlock procedure, and then you go to the computer that collects the data, and then you click the button Collect. You save your file under a certain name, and after you collected the scattering from the file, you go back into the hutch, wash the sample cell, dry it, and load your buffer. Then you exit the hutch, and you repeat the measurement for the buffer. With exposure times of 10 minutes back then, it was quite an annoying procedure. Besides, when you have many samples and it is late at night, you start doing really stupid things. Like, you carefully load your sample, then you fight these damn bubbles that are in there, and then finally you are satisfied with the result, and then you just take the syringe with your sample out and lose your sample, and you have to repeat everything again. So this is our old beamline X33 at the old synchrotron that is now shut down. So, and back then we had the MAR image plate detector where you needed just 2 minutes to get the data out of the detector. Well, actually 1 minute to get the data out and 1 minute to erase the detector to make it ready for the next exposure. The exposure time was also 2 minutes, so it was not such a big problem. But the big advantage of that beamline was that we constructed a sample changer, which is basically a pipetting robot that can pipette your sample. You can just add your samples and your buffers in Eppendorf tubes, and it will pipette your sample inside the sample cell where it is exposed to X-rays. So then and this was really a breakthrough because nobody was doing SAXS with a sample changer back then. And we had it since the end of 2007, I believe. It was not a very good solution. We got a better sample changer later. So we also got the first Pilatus detector, first outside of the Swiss light source where it was developed. And then we got an upgrade for that detector to double its resolution. And besides, we created software that would control all this hardware. So we could just say measure, and the software would control the sample changer, load our sample, make sure that the sample is loaded, and then it would control the detector and start the data collection. So on top of that, we had a user-friendly graphical user interface that would permit the user to enter all the data and to observe the measurement process. And this made possible to make the measurement process like a remote operation. So you can actually sit here in Taipei and control the beamline in Hamburg. Like you can even do it now. If we had more time, I could try to connect there, but it's late at night there. So I guess we wouldn't see much going on. But anyway, so you can actually send your samples and then control the measurement process from home. So then we even got ourselves a better beamline. So that's our new beamline P12 at the Petra 3 ring. So this beamline permits to automatically, or let's say automatically change the sample detector distance. So if you have a very large sample, you can increase the sample detector distance. And it runs the same software. So besides, we got ourselves a better, more compact, and more reliable sample changer that works with various types of samples. And the next problem we had to face is that it produced a huge amount of scattering data. And all this scattering data that we suddenly were able to collect in a very short time, it all needed analysis. So that was part what I did during my PhD studies, is automation of the data reduction and analysis. So all the steps that I was telling you, starting from radial averaging and ending with ab initio shape reconstruction, are nowadays automated. So you can just, right after your measurement, after 1 minute or so, the longest part is the ab initio modeling. After a few minutes, maybe, if you choose to have higher resolution ab initio models, you already get the shape from your protein. So this was possible using the modularized tools. So that's a number. So each data processing step is implemented as a simple program, like a program with a simple unified interface. And these programs are available for download. So if you prefer to use command line tools instead of the graphical user interface, the programs are available as part of the SAXS package. So, and this way we ended having this automated data analysis pipeline where each separate task would be abstracted in so-called components, and this pipeline is highly flexible. So if you're interested in installing this pipeline for the data, for the processing of the SAXS data, so please do contact us and we will help you to configure and install it. So the result of the data processing comes in the XML format, and it's extremely user-friendly. You can either generate Excel spreadsheet out of it, so you have all your measurements in this spreadsheet with all the parameters, or right at the beamline, or in case of a remote measurement, right at home, you see a table with a summary of the results. So this is easy to use through web, and this table has links to the generated files and models. So the beamline is also, it's possible to control the beamline through the web interface. So nowadays when we have to do user support and the user calls us at 3 o'clock at night and saying like nothing works, so typically you can take your iPhone and just log in to the web interface and see all the parameters, what is going on in the beamline, and to some extent even you can, like to a very limited extent, but you can control parts of the beamline through your phone. This all permitted to perform the world's first remote SAXS experiment. It was in Dmitry, my boss, he was in Singapore, and in May 2006 he was able to demonstrate there how he would control the beamline and actually collect the data from a protein. So I must say we don't have that many users who use remote access, but we do have some. So usually that's users who are fairly confident with what they are doing and who prepared all the samples carefully, all the concentration series. So our job is just to take it from dry ice, put it to inside the sample changer, and the user will do the rest. So I'm quite convinced that SAXS has a big, bright future, and it will become a routine method for screening of multiple samples. It's already a high-throughput technique that is available for the scientific community, and we are working on making it more smart and introducing more checks, more data validation, and to make it more foolproof and more user-friendly. And I thank you very much for your attention, and I hope that you will enjoy SAXS in the future.